Okay. So we are doing a video on protein synthesis. If I can get it to start.
Of DNA. Have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes. But in order to have that pigment, you have genes, which are portions of DNA that can code for proteins, which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription, and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for a protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You can see on the codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA.
Everyone should be logging on to Pear Deck, and your code is up. Yes. Okay.
My Chromebook's not working.
You can log on with your phone. Am I still waiting on anyone to log in?
Log in. Don't get sick.
Yeah, I'm almost there. Hold up. A-H-T. Wait, we're allowed to talk, right? We just can't. Oh,
Or be disruptive.
and are we?
Is that everybody?
Everybody knows.
That's being disruptive.
That's a disruption? That's what I'm here for.
Okay. We are doing ribosomes and protein synthesis today.
Ribosome symptoms.
Your vocabulary is polypeptides, genetic code, codons, translation, and anticodons.
Oh, yeah.
The standard is biology LS 1.4, which is demonstrating how DNA sequences inform its codon through transcriptional and translational processes. You'll be explaining how genetic codes work, describing the roles of ribosomes, and understanding how molecular biology relates to genetics. Okay. I want you guys to draw or explain two things that you guys already know about DNA and RNA.
That's what the alphabet.
Why you record yourself?
You what?
Why you recording yourself?
I'm recording the class for my class.
Oh, you're streaming.
I see a lot of people writing that DNA is a double helix and RNA is a single helix, which is correct. Is anyone still working on this one? Okay. Review from what we did a few days ago. Transcription is on the right hand side. It is when RNA goes into DNA and takes a little piece of it and duplicates it and then takes it out of the nucleus. The genetic code, proteins are made of joining amino acids together into chains called polypeptides. The 4 bases of RNA form a kind of language with just 4 letters. This is called the genetic code. And then genetic codes are read with 3 bases at a time, like what we saw in the video. So each 3 sections is a certain codon, which is a certain polypeptide as well. There are 64 possible genetic codes. Okay, you guys saw this on the video. When it starts with 3 different types, you start with this inner one, and then you move on to the second one, and then the third one, and then that will bring you to your polypeptide.
Oh.
Can we show the air on?
The arrow is on.
Oh. Sorry.
Does anyone have questions about this part?
No.
Okay. Okay. Now I want you guys to do it. I want you guys to find the names for the following sequences: AUG, CCG, and GAC. Remember, you start with the first one in the center. Remember that we should be giving a name, not a code. Almost everyone got it right. So for that's okay.
OG, GAC, and CCG.
Can't remember how to spell the words.
Should we code the code on CAC?
So for
Yeah.
AUG, you start with the A in the center, and then the second one is U, so you follow it down, and then the third one is G. So it should be the methionine is the first one. The second one is C, you start the inner one, then C, and G, so it should be proline. And then the third one starts with G, and then you go to A, and then C. So that should be aspartic acid. The methionine codon also serves as the initiation or the starting codon for protein synthesis. And then there are 3 different stopping points for codons, which in translation, my bad. Well, this one it only shows 2, but these 2 right here are 2 of the stopping points. For translation, that's what they showed in the video of the tRNA bringing it to the mRNA. Ribosomes use a sequence of codons and mRNA to assemble amino acids into polypeptide chains. This process is known as translation, which is that little picture on the bottom, which you may have to draw at the very end. In addition, each tRNA molecule has 3 unpaired bases called anticodons. The anticodons are complementary codons to the mRNA. which goes to the matching pairs like the T and the U's. So this is kind of the steps to translation. They bring in the codons, then the polypeptide chains connect together, and then the tRNA goes away, and then it's just left with the polypeptide chains. This is a picture of translation and transcription. Transcription, remember, comes out of the nucleus, which is the DNA and the mRNA. It comes out, and then it goes into the ribosome where it connects.